Well, hello everybody. Welcome to the week 3 mini review video. And this review probably will be truly mini. I don't see this going for too long because most of you reported that you didn't have all that many questions or all that much confusion on this material at all, which is great. So just a few terms that came up commonly that we'll review to start off the video. And then what most of you said is that you would like some practice problems on this material, just to kind of see how this material is applied to genetics problems. So we'll go through a few of those just as examples here, but rest assured that the challenge questions this week, of course, are all about practice and practice problems. So you get plenty of more opportunity to do some of this on your own as part of the challenge questions. The first two terms that came up as confusing, actually the only terms that we'll explicitly review, are Testcross and Backcross. So let's start with Backcross first, because in some ways it's the more obscure one, and it's one that we will rarely go back to after this week. In experimental sciences, it is extremely important for the organisms we use to be what's called genetically pure. And what we mean by genetically pure is that most of the genes are homozygotic. So for genes, they're either homozygotic dominant or homozygotic recessive. There are very, very few heterozygotic genes in these organisms. And we do that in our model organisms in the lab so that the differences that we do observe phenotypically are due to the specific gene or genes that we're studying. And there are no phenotypic differences due to any genetic differences in the background because the organisms are all genetically pure. They're all homozygotic. We also refer to this as true breeding, which is a term that will come up in your lab quite soon if it hasn't already. So we want true breeding, genetically pure, homozygotic model organisms in the lab so that the only differences between individuals are the differences we've caused to happen intentionally as part of our study. How we get there, the way that we create genetically pure model organisms, is by backcrossing. So backcrossing occurs when we take an organism and mate it with its parent, essentially. So let's say that we want a homozygotic recessive organism. So maybe we start off with two true breeding parents, and all of their offspring are going to be heterozygotic. What we want is true breeding recessive. So then what we're going to do is a backcross, and we're going to take one of those heterozygotic offspring and mate it with its parent. And in that case, we're going to get 50% heterozygotic and 50% homozygotic recessive. And we're going to do yet another backcross. And now, after the third generation, 100% of our offspring will be what we want, homozygotic recessive. So backcrossing just means mating offspring intentionally with their own parents in order to create a genetically pure, homozygotic, true breeding model organism. And again, we're not going to use the term backcross all that often because we're basically going to stay in the realm of pure genetics rather than experimental genetics. The other related term that we will use is test crossing or a test cross. This is a term that we will use quite a bit. A test cross is just that. It's a cross we do to test the genotype of an organism with a known phenotype. So let's imagine a dominant organism. And that dominant organism can, of course, be either homozygotic dominant or heterozygotic. By just looking at the phenotype of this organism, we don't yet know the genotype. And it's often important for us to know the genotype. So what we have to do is a test cross. A test cross is mating a dominant individual of unknown genotype. And the way that we're going to represent that genotype is in this way. We're going to use this kind of notation quite a bit in the course. We know that the individual has at least one dominant allele because they're showing the trait, but we don't know what the other allele is. It can either be another dominant allele, making them homozygotic dominant, or it can be a recessive allele, and they would be heterozygotic. We take that individual and we do a test cross. A test cross is defined by mating that individual with a recessive individual. This individual shows the recessive trait, and the only way they could show the recessive trait is by being homozygotic recessive. So we know this phenotype corresponds to this specific genotype. Now, by doing this cross, we have two possible outcomes. Either the dominant parent is homozygotic dominant, and the cross that we're doing looks like this, or the dominant parent is heterozygotic and the cross we're doing looks like this. In both cases, the other parent is homozygotic recessive because we know that to be the case. If reality is option 1 and the dominant parent is homozygotic dominant, then we would expect all of the offspring to be heterozygotic, meaning 100% of the offspring should show the dominant trait. If, conversely, the second option is what's really the case, and the dominant parent is heterozygotic, we would see this outcome, and 50% of the offspring would show the dominant trait, and the other 50% of the offspring would show the recessive trait. So the Testcross shows us what the second allele is in the dominant parent by looking at the offspring and using that offspring to inform us as to the genotype of the parent. So if we see any recessive offspring after the Testcross, then we know that the initial dominant parent was heterozygotic. If every single offspring we observe in the next generation after the Testcross is dominant, then we can assume that the dominant parent was homozygotic dominant. So that's a Testcross. All right, the next thing I'd like to do is tie what we have done this week with Mendelian genetics to what we learned last week about independent assortment in meiosis. And it really goes back to what we have just been saying. So genetically, from a Mendelian perspective, if two heterozygotic parents mate, we represent it as a Punnett square, and we have these outcomes that are possible. Genotypically, we have a 1 to 2 to 1 ratio, homozygotic dominant to heterozygotic to homozygotic recessive. That's a genotypic ratio. Phenotypically, we have 1, 2, 3 dominant individuals to every 1 recessive individual. That's our phenotypic ratio. And that's all Mendelian genetics. Basically what we're saying is if we have a heterozygotic parent over here, they can contribute to their offspring either a dominant allele or a recessive allele. If we have a heterozygotic parent here, they can contribute either a dominant allele or recessive allele. All of those alleles are of equal likelihood, and so we get these proportions of offspring. If we go at this from a meiosis point of view, and I'm going to capture these cells in metaphase 1. These are sister chromatids, so they're going to be identical. And this is the homologous pair. These are sister chromatids, so they will be identical, and this is the homologous pair. If you fast forward in your mind's eye through anaphase 1, which is going to pull these chromosomes apart, and then go through meiosis 2, these sister chromatids will eventually become their own individual chromosomes, and we're going to have gametes that contain a dominant A, 2 of them in fact, and we're going to have gametes that contain a recessive A, 2 of them. And here as well, we're going to have a dominant A in 2 of these gametes, and we're going to have a recessive A in 2 of these gametes. An equal number of dominant and recessive gametes in both parents, and an equal likelihood of contributing them. That's why these Punnett squares work, because they represent that equal likelihood. All right. So what's going on in meiosis dictates the alleles that a parent can contribute in sexual reproduction, and that's what we're trying to track with Mendelian genetics. Let's go one step further. Now, let's think about a dihybrid cross, where we have 2 heterozygotic parents that are heterozygotic for 2 different genes, A and B. And though I despise it, we could represent this as a 4x4 Punnett square. We use the probability method in this class, but we could represent this as a 4-square Punnett square. In other words, there's an equal likelihood of the parents contributing each one of these alleles independently of any of the other, and we have to account for all 16 possible outcomes. That's really what we're trying to represent here. From a meiosis perspective, as long as these genes are unlinked, meaning they're found on different chromosomes, we can represent what happens in meiosis in this way. If these chromosomes happen to line up in this orientation during meiosis 1, metaphase 1 of meiosis 1, and again, you fast forward in your imagination through anaphase 1, where homologous chromosomes are pulled apart, and then through meiosis 2, where sister chromatids are pulled apart. In this case, because these chromosomes are on the left and these chromosomes are on the right, and these chromosomes are on the left and these chromosomes are on the right, in this case, we will get 4 different kind of gametes. We will get A and B dominant together, A and B dominant together. That's these 2 sister chromatids and these 2 sister chromatids. Recessive A and B together, recessive A and B together. And that's going to be the case over here as well. But independent assortment says that where the maternal chromosome goes has no influence on where the paternal chromosome goes, and that different chromosomes can go in different places arbitrarily and randomly. So just as likely as this scenario are chromosomes lining up like this in metaphase 1. If chromosomes line up in this way in metaphase 1, we get a completely different set of gametes. Again, fast forward in your mind's eye through the rest of meiosis 1 and meiosis 2. We have 4 possible gametes that we can make. We can either make dominant A with recessive B, and we'll get 2 of those, or we can get recessive A with dominant B, and we'll get 2 of those. Because these genes assort independently, because they segregate independently during meiosis, we get equal numbers of all 4 different gamete types. And it's because of that equal likelihood that we represent all 16 possible outcomes as equally likely with one another. So what happens in meiosis, especially in meiosis 1, dictates specifically the way that we do the math in the Mendelian genetics. So let's go back to the true Mendelian genetics that we want to be doing when it comes to dihybrid crosses. Let's cross these 2 organisms and see what we can get phenotypically. Well, if we just kind of rattle our minds, what we can get phenotypically is really just 4 possible outcomes. Either the organism is dominant for the A and B traits, the offspring is recessive for the A and B traits, the progeny that we get are dominant for A and recessive for B, or the organisms we get in the next generation are recessive for A and dominant for B. Those are the only 4 phenotypic classes we can possibly have from this cross. In order to calculate the probabilities of all of them, we will not use a 4x4 Punnett square, we'll use the probability method. We're going to break the 2 genes down, and we're going to just take this cross 1 gene at a time. We're going to diagram the A cross first, which is a heterozygotic mated with a heterozygotic, and we're going to diagram the B cross independently, and that's a heterozygotic by a heterozygotic. We'll fill in those Punnett squares, and these are just mini monohybrid crosses, so this is nothing all that heavy, things that you've seen before. And then we're going to use these individual Punnett squares, these individual crosses, to allow us to calculate these probabilities. So, what is the probability that an offspring is going to be dominant for A? Well, this will be dominant, this will be dominant, this will be dominant. That's 3 out of the 4 possible options. What is the probability that this offspring will be dominant for B? 1, 2, 3. These are all dominant here, over a total of 4. Now we have those 2 individual probabilities. We need to think a little bit semantically here in the English language, because what we're really asking is, what's the probability that an offspring is going to be dominant for A, here it comes, and dominant for B. And once we have that and, we're going to know that we're using the product rule. We're going to multiply. When multiplying fractions, you multiply the numerators and the denominators individually. So 3 times 3 is 9, 4 times 4 is 16, 9/16 of the offspring are going to be dominant for A and dominant for B in this cross. What proportion of the offspring are going to be recessive for both? Well, we have to go to our independent individual probabilities. We have well, that's not recessive, this is the only recessive one, so that's a 1 in 4th chance for A. These are not recessive, this is the only recessive here, so that's a 1 in 4th chance for B. We're looking for the probability that an offspring is going to be recessive for A and recessive for B. 1 times 1 is 1, 4 times 4 is 16, that's a 1/16 probability. How about dominant for A and recessive for B? Well, dominant for A is a 1, 2, 3 out of 4 probability. Recessive for B is a 1 out of 4 probability. We want the probability of them both happening together, that's an AND event, that's 3/16. And finally, the probability of being recessive for A is only 1/4. The probability for dominant B is 3/4, and that's also 3/16. So this is the classic dihybrid proportion. It is a 9:3:3:1 ratio, a 9:3:3:1 ratio, phenotypically. That's a phenotypic ratio. And what is it a ratio of? It's a ratio of both dominant to one dominant, one recessive, to the other dominant, one recessive, to both recessive. So it's a 9:3:3:1 ratio of these individual phenotypic outcomes. That's how meiosis and independent assortment leads to the principle of segregation in terms of Mendel's genetics and brings us to how we approach dihybrid crosses when those genes are unlinked and present on 2 separate chromosomes. So now let's go on to a couple of practice problems. Just kind of quick practice problems, not really well formulated, and certainly the wording is not going to be ideal for these practice problems. Your real practice problems, your challenge question problems, your quiz questions, your exam questions, they'll all be well worded and well formulated. Here we're just going to focus on the genetics. So let's say that one problem says to you that you have an organism that's showing the dominant phenotype. You mate it with an organism that shows the recessive phenotype. And when they mate, 50% of their offspring show a dominant phenotype and 50% of their offspring show the recessive phenotype. And perhaps the question asks you, what are the parental genotypes? I'm writing phenotypes there. But what are the parental genotypes? In other words, what genotype do these two parents have? Well, a couple things you can do right off the bat. You can say, well, if an individual is recessive, they only can have one genotype. They have to be homozygous recessive. That's the only way you show the recessive phenotype is by being homozygotic recessive. For the other parent, they're showing the dominant trait. So we know they must have at least one dominant allele, but we don't know what the other allele is yet. So we'll leave a placeholder there. And of course, the question says they're crossed. So what you realize right away is this is pretty much a Testcross. If this individual was homozygotic dominant, then all of the offspring from their mating should show the dominant trait. And that's not what we see. Half of the offspring show the recessive trait. So the only other option is that the dominant parent is heterozygotic. What we do to check our thinking is we diagram that cross. So there's the heterozygotic parent that we think is heterozygotic. There's the recessive parent showing the recessive trait. We diagram the cross as a Punnett square. And we see that indeed, given that cross, we would expect 50% of the offspring to show the dominant trait, and 50% of the offspring should show the recessive trait. That's exactly what we say we saw, and so that's the correct answer. So that's like an inference kind of question, where you're using offspring to infer what parental genotypes might have been. Let's go to another kind of question. In this question, we could ask a numbers-based question where we're using the math. So maybe we're told that 2 dominant parents are mated, and when they mate, they have 40 recessive offspring. And this would be a 2-part question. The first part would be, what are the genotypes of the parents, and the other would be how many total offspring did they have. So we have an inference question and we have a math applied to genetics question. I'll walk you through both. So if these are two dominant parents, parent number 1 should be represented like that, and parent number 2 should be represented like that. We know that each of them must carry at least one dominant allele because they're showing the dominant trait. But we don't know what the other allele is yet, so we're going to leave a placeholder there. And we're told that of their offspring, 40 of them are recessive. Well, in order to be recessive, this allele had to have come from one of the parents. That means this parent has to have a recessive allele to give. By the same logic, this allele had to come from one of the parents, and that means this parent had to have a recessive allele to give. Now, one of you, or a few of you out there might be thinking, well, if that's the case, how do we know that one of these parents wasn't homozygotic recessive, and they had 2 recessive alleles to give? No. We were already told that both of these parents show the dominant trait, so they have to have at least one dominant allele. And in order to give rise to an offspring that has 2 recessive alleles, each parent had to have a recessive allele of their own to give. So those parents have to be heterozygotic. So what's the genotype of the parents? Both of them are heterozygotic. That's how we're having recessive offspring. Now we diagram the cross. There's one heterozygotic parent. There's the other. We fill in the Punnett square, and here are those recessive offspring. That's what explains the 40 that we see here. So 40 offspring are recessive. This is a Punnett square. It's meant to show that 25% of the offspring fall into all 4 of these classes equally. That means 40 offspring are heterozygotic here, 40 offspring are heterozygotic here, and 40 offspring are heterozygotic here. The number of offspring should statistically be equal in all 4 boxes. Since only one box has recessive individuals, that sets the box value as 40, and the others have 40 as well. So how many total offspring did they have? Well, if we count them up, we see that they had 160 offspring. So that's another example of a kind of question we can do with this material. The last one I want to do is a multigene cross. And as I promised you in the lecture, if you use the probability method, you can diagram and predict a cross using any number of genes you want. That gets to be very, very unwieldy after dihybrid crosses with the 4x4 Punnett square. That's why we're never going to use them. So let's make this a little bit straightforward and just give you some parental genotypes here. So we had a 5-gene cross. Now you'll see when letters are the same, I underline them. That's so that we don't get them confused. This is a tip I usually pass on in class. So some letters are very distinct in the differences between their capital and their lowercase. And for there's no confusion. Other letters, not so much. O's and C's are good examples of that. And when you're in a rush, especially when you're handwriting this, and you're kind of going very quickly, and you're trying to do heterozygotics and homozygotics, it's easy to forget what was what when you go back to your notes. Did I mean homozygotic dominant or homozygotic recessive here? If I put 2 O's down, is this 2 big O's, 2 little O's? Is this a heterozygote? Things get messy when you're in a hurry. So I've gotten into the habit, and I recommend you do too, that whenever letters are similar in that way, you put a little underscore under the recessive one. So that even if your letters get to be a little bit ambiguous, and it's getting harder and harder to tell, you know that this is homozygotic recessive, and this is homozygotic dominant because of your underscores. So that's what I'm doing up here. All right, so now let's ask, what is the probability of these 2 individuals mating for all 5 genes, and they have offspring that are dominant for A, recessive for B, dominant for C, recessive for D, dominant for E. These are phenotypic classes. So what is the probability that an offspring from this mating will be dominant, recessive, dominant in their phenotypic traits? Well, I could do what would it be? So it would be, I could do a 10 by 10 Punnett square, giving me 100 different possible outcomes, and be here for the next 3 hours with this video with you. We could do it that way, but we won't. But we could also use the probability method and just break these genes out and consider each of these crosses one gene at a time. So I'm going to put down 5 different Punnett squares. I'm going to fill these Punnett squares in. And while I do that, I'm going to fast forward the video so you don't have to sit here and watch me do it. So here I have the Punnett squares all laid out. Let me get rid of this arrow because it's getting in the way. And now we'll fill in the outcomes of those individual crosses. So each of these Punnett squares has now been filled in based on the crosses that were indicated, based on the individual genes that were indicated. So all of the data we need is right here now. So what we have to do is go back to our prompt and go to our Punnett squares and see what the probability is for each of these individual phenotypes. What's the probability of being dominant? Well, it's 100%. All those offspring are dominant for A, so that's going to be a 1. What's the probability for being recessive here? Well, half of them are recessive and half of them are dominant, so that's a half. What's the probability for being dominant here? Well, that's a half. What's the probability of being recessive here? That's a half. What's the probability of being dominant here? That's a half. And what's the probability of being dominant for A and recessive for B and dominant for C and recessive for D and dominant for E? Well, that's all of those multiplied. 1 times 1 times 1 is 1. 2 times 2 is 4 times 2 is 8 times 2 is 16. That's a 1/16th chance of getting this particular gene phenotypic combination from this multigene cross. So you can see it's not unwieldy. If you understand monohybrid genetics, then you can do the genetics of any number of genes that I throw at you, as long as you break those down into individual monohybrid crosses and use your probability to ultimately predict the probability of any particular kind of offspring given that mating. So that's pretty much all that I have for you today. Again, not much in terms of review because all of you are rock stars and pretty much felt like much of this was clear, but you did want that extra pass through the math. You did want that extra pass through the genetics, through the lens of what kinds of questions could you be asked, and that's what I've tried to provide here. As we go into the challenge questions, you're going to see the questions there are quite similar in their construct to some of what we've talked about here. That'll give you a chance to practice this on your own. And then, of course, the week 3 quiz will be the ultimate assessment of how well you can do these problems on your own and unaided.